Okay, so as I recall, we did finish chapter 3 lecture slides. I think that's right. So we talked about potassium channels, and really there's a way in which chapter 3, I mean, like the points of chapter 3 are to understand how movement of ions can cause membrane potentials, and therefore if you change which ions are moving and which direction they're moving, that'll change the membrane potential. We also sort of established that in a resting neuron, we're dealing with membrane potentials that are typically very negative. What we're going to be talking about in this chapter is basically what happens when a neuron gets excited. It will fire what's called an action potential, which is a radical, massive change in membrane potential that occurs over a short period of time. So that's what we're going to be trying to understand. In this chapter, we have sort of just a general introduction to action potentials, just so we have a feel for like how we think the information is coded within them. But then really most of the chapter is going to be focused on understanding in molecular level mechanistic detail what exactly is happening during an action potential. So how exactly it is that they work. So we'll go through sort of a theoretical action potential and then talk about this key structure called a voltage-gated sodium channel that's responsible for them, and then add in some additional details about what happens in real life, not just this theoretical one with action potentials and how they work. So again, the idea is that we talked about resting neurons in the last chapter. Neurons are interesting in a lot of ways because they're electrically excitable. And so they transmit signals by having changes and disruptions in their resting membrane potential. So for example, I'm going to dim the lights just because I can't feel like I'm getting to see anything. So you probably don't either. If we step on a thumbtack, which is what this poor individual is doing. So here he is strolling happily along. And he's about to step on a thumbtack. When he does so, that thumbtack is going to stimulate various receptors, pain receptors that are present within his or her foot. I feel like that's a his, but I don't know. It doesn't necessarily have to be. It'll stimulate pain receptors in that person's foot, which will then be relayed up axons that travel all the way from his foot up into at least his spinal cord, and in the case of some of the touch neurons, just providing touch reception, which we can feel the thumbtack going in addition to feeling the pain, those will go all the way up into his brain as well. So the way in which they are sending signals, or the way in which the system works basically, is that the pain receptors have various receptors that allow them to perceive that cells have been damaged around them. And that's going to happen with the dendrites of these pain receptors. If there's sufficient stimulus on those dendrites, and the stimulus, I mean, to detect damaged cells, a lot of times you're detecting either extreme mechanical pressure and force, or you're detecting, a lot of times, like the chemicals that are released, the compounds that are released from cells that are not normally found outside of cells. Once those are detected, that stimulates pain receptors. If there's sufficient stimulus, that will result in the axon hillock of these pain receptive neurons to reach threshold, which will then trigger a barrage of action potentials that will propagate and travel like little waves up this person's leg, up into their spinal cord, and then usually through relays of additional neurons up the spinal cord, up into the brain, to the point that you can actually perceive that you just stepped on the thumbtack. These action potentials are massive disruptions in membrane potentials. So if you're recording from one spot on the axon, this is what you would see with one action potential going by. You would start at your nice negative resting membrane potential. So you can see in this graph, this is time on the x-axis. This is your membrane potential on the y. We're starting at about minus 65. And then what will happen is there'll be an initial stimulus that'll get us to threshold, which is just a little bit above minus 65. And it's not really obvious that there is this initial stimulus in this figure, but we just have to infer it. But then, in this one spot in the membrane, you'll have a massive upward deflection in membrane potential. So we're going from minus 65 millivolts all the way up to like plus 35, almost plus 40 in this case, on this picture for this neuron. And then, immediately, we have this massive repolarization. We shoot right back down. And we even overshoot. So, you know, here's rest right at minus 65. We go under that resting membrane potential and then work our way back up. So we have this sort of massive up, massive down, to the point that we kind of go under a little bit, and then we make our way back. And the whole thing is over in about 3 milliseconds. So it was this really fast up and down. I mean, that's almost instantaneous. Not quite, but it's almost instantaneous. You can't just see it on a recording unless you really stretch out the axis.
Most of the time, if you're just looking at recordings, you just sort of see a little line go up and down. And it's just a virtual, absolute straight line most of the time, unless you really blow up the scale of the time axis. So our job in this chapter is to try to understand how this thing worked. Like, what exactly is happening there? And we'll kind of do it qualitatively as well as quantitatively by applying that golden equation to this process. In terms of what is that actually doing for us in terms of information, because the whole point of these action potentials is that they're relaying information in some way. I think that's really nicely illustrated in this particular experiment. So in this experiment, what they've done is they've dissected out a frog gastrocnemius muscle. So this is, this muscle in the back of the frog leg. This is what everybody wants to eat when you get frog legs, if you're into frog legs. And they're suspending that from a solid surface. So they've got a nice firm wire here on one end of it. But on the other end of it, they basically have the ability to apply force. And then what they've done is that in addition to dissecting out the muscle, they also dissected out the nerves that would relay touch information, stretch information coming out of this muscle. So these are being recorded by little electrodes that are placed here. So you can kind of make out these little electrodes that are embedded into the muscle. Really what they've done is they've embedded those into the nerve that's conveying that information. So these are a collection of axons that you're recording from that are conveying stretch information. And then, in this experiment, what we do is we basically just hang different weights off the end of this frog muscle. I said from a frog leg. It really is, it's the muscle from a frog leg. So we could hang a 2-gram mass off the end of it, or we could hang a 5-gram, or 10-gram, or 20-gram mass off the end of this muscle. So that's going to stretch the muscle by different amounts of force. And we're recording from sensory neurons that record stretch. And here's what you see. So here's, there's sort of a baseline recording, and there's a lot of noise down this baseline recording. That's just the way they are. But every time you see one of these little blips, beep, beep, beep thing, these things, those little blips are an individual action potential. And you cannot see it going up and down because it happens so fast. They just look like a perfect vertical straight line. Because our scale, in this case, do they give a scale? I mean, the scale is probably more like, this is a couple seconds worth of a recording. But we can't see millisecond up and downs. So we just see a vertical line because the up and the down happens before any real time passes. But you can see a difference here, right? So if we go from 2 grams to 5 grams to 10 grams to 20 grams, what are you seeing that's different in these recordings? I mean, we're just sort of suspending a weight off these guys. But what does look different? Yeah, so we've got, if you look at the 20 gram versus the 2 gram, and these are all intermediate in here, going from 2 grams to 20 grams, we have much higher numbers of action potentials being fired. If you look at the actual action potentials themselves, they're pretty much the same, right? And even within the 20 grams, I mean, there's a little variation in the up and down. That's probably just random noise. I mean, there's always electrical noise when you're recording at this level. You have to amplify signals quite a bit to detect them. For the most part, the action potentials are exactly the same throughout this entire recording, and the ones here are the same height as the action potentials up here. There's not really a difference across these in terms of how high each action potential is. The action potentials, each one is kind of the same as every other action potential on this figure. What's different is how many you're firing per second. So when there's a very small stimulus, in this case a small sensory stimulus, there's very few action potentials being fired. There's occasional, like here and there, and there's a little bit of weight that's being suspended there. If you took off all the weight, there still might be 1 or 2 that you detect. But as you add additional weight, you progressively see higher and higher action potential frequencies. And you would eventually get to a point where you hit kind of a maximum, where there's the maximum number of action potentials per second that's being conveyed up that axon. If it works, well, here it is. So this sort of summarizes this, that we're seeing an increase in frequency of action potentials. So the frequency is going up, the number per second, but the size or the amplitude, so amplitude is like, how large each wave is, each action potential surge, all the amplitudes are the same. Every action potential, every individual one is the same as every other one. If the sound thing works, I've got a couple recordings here of just if you kind of hook this up to a speaker such that the speaker just makes a little white noise every time there's an action potential pulse, you can listen to action potentials. So a lot of like old-fashioned electrophysiology labs would do this.
Let me see if this works. Oh, come on. Let me try again. Really? Oh, you know what? I think I know it. It's like, how many volume recordings do I have here? I've got 4 or 5 different switches. Let's try again. Oh, this is really sad. Well, okay. I'm about to give up. Oh. Okay, so we heard that. So that is not a cricket. What you just heard is literally a recording where they're recording from sensory neurons that are in somebody's tooth. And somebody's going tap, tap, tap on the tooth. So I'm going to play it again. And so you can kind of hear when you hear that. It's not just like do, do, do. It's like a bunch of pulses. Maybe if you hear like really closely. It's a bunch of pulses in a row, and then it stops. And a bunch of pulses. So for each tap, there's like a barrage of action potentials that's been released. It sounds like a bunch of clicks. And then there's another one that's more like constant pressure placed on the tooth. So that's this one. All right, so I'll just play that the one time. But that's, again, that's like this barrage of action potentials. Each one is causing a little bit of white noise, and it results in this kind of clicking sequence that's going through. It's not that our nervous system listens to these signals, but I think it does kind of give you a feel for what these signals would actually sound like if we were able to sort of listen to them. I like listening to them. Like I said, there is usually an upper end to how frequently you can fire action potentials. So usually it's not possible to have more than one action potential occurring per millisecond. That's kind of their upper limit for most neurons. That's referred to as this absolute refractory period, where you can't go and have the absolute refractory period is like this period of time after you start an action potential that you cannot start another one. And usually it's about 1 millisecond, which means you can't have an action potential closer than about 1 millisecond to the next one. That's kind of your upper limit. And that kind of makes sense, because if we go back here, like this whole, an action potential itself, we started on this graph, we're kind of starting the action potential about a half a millisecond, and we're at least down to this point by about 1.5. So this whole thing from here to here, that takes about a millisecond. So you really can't have more than one of these overlapping at a time. They kind of, and there's a mechanistic reason that we'll talk about for why that is, but each one will tend to hold by itself within a neuron. Otherwise, you just have like a constant depolarization, and you wouldn't have the up and downs anymore. Oops, sorry. Okay. Okay, so if we look at one action potential, we can break down the different parts of an action potential into different phases, and there's different books that'll define these in different ways, but this is pretty standard. So here we are down at the resting memory potential. There'll be a rising phase for an action potential, so that's going up this way. I call this a depolarization. So that kind of makes sense if you sort of imagine we're starting polarized. So 0 millivolts would be neutral. So at rest, we are polarized such that we have a lot of potassium going out, potassium leak channels. The inside of the membrane is fairly negative, about minus 65-ish or so. And so by going up towards neutral, we're becoming less and less polarized. So this is called a depolarization. Now, technically, we overshoot, right? But I don't know, just by convention, most of the time, when the memory potential is getting more positive like this, we call it a depolarization. At least that's what I always do. So we have a depolarization that goes up into this overshoot. We overshoot 0 millivolts usually to maybe about plus 30 or so. And then we come back down. We have a falling phase of an action potential that results in a rapid repolarization. So we start up here in the positives, we go past our 0 millivolts, and then we repolarize back down to this highly polarized negative value. And in fact, we actually undershoot. So we go past resting membrane, which is shown by the dotted line here. We go past the resting memory potential into this hyperpolarized area. So it's extra polarized. And then eventually, we return back down to our resting memory potential after 3 or 4 milliseconds or so. So the whole thing is at least going from about here to maybe about here. Really, that's about 2 milliseconds or so. If you include some of the sort of initial things that happen, you can get into like the 3 millisecond range. That's about where you are in a typical neuron for a typical action potential. different neuron action potentials will look a little bit different from the others. Okay. So that's, the thing that we're trying to study is these action potentials. So our goal then is to put the pieces together and mechanistically understand exactly what is happening during this action potential. So how is it that we can make action potentials and create them? That's our goal. So if you think of what we just did, it's kind of like we did an experiment. We recorded what's going on. We can witness an action potential. We see this up and down. We need to then figure out an explanation for how it is that these things work. How do those cells achieve that? So we need to have neurons that are able to respond to some initial stimulus. Like we said with the pain receptors, it's got to be some kind of an initial stimulus. And usually that stimulus is going to occur out on a dendrite. And then somehow that signal needs to get from the dendrite to the action potential. We're going to kind of just assume that it does for now. When we get to the next chapter, chapter 5, we'll be looking at synapses. And when we talk about synapses, we'll talk about how we get from the synapse on the dendrite back down to the axon hillock of the next neuron in the sequence. But for now, we'll just sort of assume that that happens. We'll talk a little bit about it, but not much in this chapter. Once we get to the action potential, or get down to the axon, and we start making an action potential, we need to figure out some way that action potentials can feature a massive depolarization. So we need to have a massive positive increase in the memory potential. And it needs to be done in such a way that each action potential basically has the same amplitude, such that every action potential is the same as every other one within a neuron. Then we need to very rapidly repolarize and get back down to our resting memory potential. And then we also need some mechanism to prevent stacking, such that each action potential sort of stands by itself. We need to have absolute refractory periods where you can't start another another action potential until the prior one is completed. So we'll have to talk about what are the mechanisms that would allow for an absolute refractory period. And there's also something called a relative refractory period that we'll talk about too. And finally, we need to have some kind of mechanism for how this could be propagated like a wave down the axon and basically regenerated along the way. So let's kind of start up at the top, and we'll sort of work our way through these requirements. So the first thing we need to do is we need to have some kind of, assuming we have a stimulus that comes in and is providing a stimulation of this neuron, how then are we getting some kind of a change in memory potential? And what chapter 3, I think, very nicely showed us is that memory potentials are caused when ions move across a membrane. So we had Ohm's law that we briefly mentioned. This is one depiction of Ohm's law. So this is saying that the current is going to be across, this is ions moving across a neuron, is going to be influenced by the conductance. That's how easily they get across the neuron. Plus something about the voltage. And this is this ionic driving force, you might remember. So this is the membrane potential currently minus the equilibrium potential of whatever ion we're interested in. In this case, it'd be, in this figure, it's going to be potassium. So what all this means is that we will get a current of ions. So potassium ions will move if they have permeability. So that's this factor. So they need to be able to get through the membrane. So there's got to be some channels for them. And if there's a difference between the current memory potential and the equilibrium potential of potassium, because if our memory potential is the same as the equilibrium potential of potassium, at that point, potassium doesn't move in a net direction across the membrane. The concentration gradient would be balanced by the electrical gradient. All that stuff we did last chapter. So in the picture, what you can see is that initially, we've got a membrane where there are channels, but all the channels are closed. So at this point, if we're focusing on potassium channels are all closed in this sort of fictitious membrane. So that means the permeability, or the conductance, of potassium is 0. So G is 0. Which means that because there's no way to get through, there are no ions moving through. We have no current for potassium. So if G is 0 in Ohm's law, then the current is going to be 0. That's what this is showing. In this picture, what we've done is we've opened the potassium channel. So now there is a permeability. So G, the potassium conductance, is greater than 0. It does have a conductance. Could be 1, could be 20. And so as a result, now we have a current. Potassium ions are flowing across the membrane. So if G is greater than 0, that means now our current can be greater than 0. And it will continue to flow until such time that we have so many potassium ions that have exited And therefore, we're building up some positive charge on the outside of the membrane, which is balanced by this negative charge that we measure on the inside of the membrane. And ions will flow until such time that the amount of negative charge inside the membrane equals, or is enough to pull the potassium ions back in at the same rate they were leaving down their concentration gradient. That's what's depicted here. So here we've hit an equilibrium where potassium ions will go from inside to outside down their concentration gradient, but they're also going from outside to inside as they're attracted to the negative charges on the inside of this membrane. And so at this point now, we have no net current either, right? Because the membrane potential, this difference between the membrane potential and the equilibrium potential is now 0, and therefore you will have 0 current. There's no net current flowing across this membrane. So all of this is basically, the intention of this is showing that when you have net movement of ions, that's going to cause some disruption and change in the membrane potential. So we should be able to cause changes like a depolarization phase in the action potential by allowing ions to flow across the membrane. Okay. So let's apply this to and create this little model of a neuron. So in this case, we'll start with getting an action potential started. This is sort of our initial stage. We said we have sensory neurons. It doesn't have to be a sensory neuron, but let's just go with that for now because it makes it easy. These sensory neurons have dendrites that are collecting some kind of a signal. When they collect the signal, like, oh, there's chemicals in here that are released from cells that have been destroyed, and that's going to indicate pain is happening, that will result in what's called a generator potential, where usually, literally what happens is there'll be some little channels that will open that will allow new sodium ions to start coming into this neuron. So I'm sort of depicting that over here. I've got it animated. So here's a little sodium ion. This will be a channel. This channel will open when this pain receptor neuron, in this case, detects that there's some kind of painful event that has happened nearby. So it binds ADP, which you don't usually find outside of cells, but there's cells nearby that broke apart, so it released that compound. Now sodium ions start rushing into the interior of this cell. And they're rushing in on the dendrite. So as they come into the interior, they're going to cause the dendrite out here to become more positive. But it's only really happening here. This is this generator potential that's happening out on a dendrite. What will happen, though, is that some of the sodium ions that come in here, some, not all, like some are going to diffuse the wrong way, some will leak back across, but some of them will make their way down to the axon hillock over here. So I'm going to do that again. It didn't quite work. I tried to keep it inside the dendrite, but I couldn't do it. So again, we're going from the dendrite. Some of these charges will literally just diffuse their way down the dendrite into the axon hillock. And in that way, they can provide some kind of a disruption to the membrane potential in the axon hillock. Again, most of them won't make it all the way down there, but some of them will diffuse down. Once that happens, if there's enough of them that make it down into the axon hillock, we will then cause enough of a positive membrane potential in this area to start opening a voltage-gated sodium channel. I'm calling a voltage-sensitive sodium channel here. So these are channels that will open. They're sensitive to changes in voltage around them. And when the membrane becomes more positive around them, these little channels, which I'm showing you one of them in blue, will start to open. When they open, they may allow additional sodium ions to come in. So here's some different sodium ions that we're circulating around over here. They may start to enter into the interior of this axon. And when that happens, that's going to cause a big change in the membrane potential in this spot out here on this axon. Okay. If there was not enough sodium ions that got from the dendrite down to the axon hillock, we would not have sufficient numbers of them to reach threshold. If we don't reach threshold, that means that we don't open very many of these sodium channels. You need to open a sort of a population of these voltage-gated sodium channels to get the process going. Once you do, though, we have this sort of inevitable massive chain reaction where we send an action potential that is produced here, and then it kind of reverberates or is propagated all the way down the axon. So it's sort of, we're going to be really focusing on what's going on with these voltage-sensitive sodium channels. So here they are in this sort of depiction that's not, this is kind of like what we saw 2 slides ago. So one way that we could make something that would look a lot like an action potential is we could first open a bunch of sodium channels like we just kind of mentioned, and then we could close them. And the result is something that looks a lot like an action potential. So here we are. So here we are at minus 80. We'll just say we're down very negative. And at this point, the only ions that can flow across this membrane are potassium ions. So we're at this resting membrane potential. Potassium ions flow out. They make the outside of the membrane more positive. Inside becomes more negative, and they would change the membrane potential until we hit this equilibrium potential of potassium. So at that point, we're at the equilibrium potential of potassium, very negative. Then what we're going to do is we're going to cause a bunch of voltage-sensitive channels to open for sodium. So these are channels that only allow sodium ions to go through. So here are these sodium ions that start to rush in. They'll make the inside of this membrane more positive, and as they do so, if we measure the membrane potential, we'll see the number going up. It'll become less negative, it'll cross 0, and eventually it'll hit a very positive value. And we've increased, we've opened massive numbers of sodium channels. It's not like we've just opened one. We've opened, thousands, if not millions of these things in this one little patch of membrane. And then what we could do to get back to rest is we could just close those channels again. So if we close those sodium channels that we had just opened, at that point, we're starting here up at this peak. We had a pretty positive membrane potential because we opened a bunch of sodium channels, but now we've closed them all. So what will happen is potassium ions will start, or will continue to flow down their concentration gradient. As they do so, they're going to make the outside of the membrane more positive and less negative. The inside is going to become less positive and more negative, and they'll flow, and they'll flow, and they'll flow, and they'll flow, until finally we get to this point that the membrane outside becomes very positive, so you're repelling potassium. The inside of the membrane becomes very negative, we're attracting again, and at that point we basically have decreased our membrane potential back down to rest. So all we did to make something that looks a lot like an action potential is we opened sodium channels here, and then we closed them again. And the result is we went up, and then we went back down again. We kind of did this on the homework even. Like we in question 3A was at rest. Question 3B was opening a bunch of sodium channels. And we went up, and then if we close those channels, we go back down again. So by themselves, voltage-gated sodium channels are able to do a lot of what we need to have happen for an action potential to exist and function. It's kind of the take-home here. So this is kind of theoretical, and this is very much what electrophysiologists were doing, is they're trying to understand these action potentials that they're recording. Is they're trying to all right, so how could this work? Well, what if we had sodium channels? Once they had an electrical model that made sense, and this is sort of what this is an electrical model that kind of makes sense, it matches what they were recording. The next question is, let's try to go find some sodium channels. And you can do this. In fact, they've done this. Literally, they've tried to record across individual sodium channels. And there's a particular method, it's called a patch clamp, that allows you to do something like this, where you could record membrane or ions flowing through one individual channel. It's a cool little, I think it's a neat method. It's technically very difficult, but I think conceptually it's not real hard to understand what you're doing. You basically have to first create a very, very small electrode. So this is basically like a neuron. And this would be a neuron in cell culture. So, or actually, honestly, this is probably not even a neuron, but let's pretend it is. It probably is a frog oocyte, to be honest with you. But what they've done is they've put a glass electrode up against it. Glass electrodes are just hollow little tubes that you fill with saline solution, and then you hook them up to wires that are literally hooked up to amplifiers and other kinds of things so that you can record electrical signals. They have to be really small. So what you do is you take, if you've ever done like a hematocrit thing where you've poked your, somebody pokes you or you've poked someone else, and then you fill up a little tiny tube with blood. You take a tube that looks a lot like that. You take it to a Bunsen burner and you heat it, and then you stretch out the glass and you just kind of go like this. And if you do it right and you're good at it, what you'll do is you'll create this very long, very thin little tube that you then take off heat, let cool, and then you break it, and you get this extremely fine little tip that is hollow inside, and you can fill that with saline solution. So what you do then, this is the original way that you do it. There are much more sophisticated ways to do this now. But then what you do is you kind of snuggle that electrode with a micromanipulator. You're looking through the microscope. You snuggle it right up against a cell that you're interested in. And then you apply the tiniest little bit of suction to your electrode. So you suck out a tiny little bit of solution from in here. And so what that ends up doing is it pulls a tiny little bit of membrane into your electrode, just like this. And if you're lucky, that tiny little bit of membrane that you pulled into your electrode might have a single channel that might be of interest to you. This is kind of a trial and error thing. I mean, you try it a bunch of times, and eventually you get lucky and you get the channel that you're shooting for. But then the great thing is, once you get one, you can record movement of ions through that channel, because this is all hooked up to your electrical recording apparatus. This is a recording from an individual, basically, of ions flowing through an individual sodium channel. So initially, there's no current at all. That's what this is showing. And then every time you get one of these dips, what's happened there is this sodium channel is open. Here's a nice big opening. And there's another nice one. And then there's a huge one here, another decent one there. So every time you get these downward deflections, what they're recording is literally sodium ions flowing from outside the cell to inside the cell through that channel. So they're getting, inward sodium currents through that one little channel. So it is possible to actually record from one individual sodium channel, and they were able to do this to help localize in real living cells these kinds of channels that they sort of predicted must exist based on the electrophysiology. And the cool thing is you don't have to do it for just one sodium channel. On one given cell, you could, if you're good, you could record from several different sodium channels all at once. And so that's what they've done in this picture. There's another technique that's mentioned here. I don't want to get into it. It's called a voltage clamp. It's different from a patch clamp. The point is they can manipulate the memory potential by just basically injecting current into the neuron. And then they can use patch clamp recordings to record from the individual channels. So these are patch clamp recordings of individual channels. And so in this experiment, what they've done is they've started with a membrane, a neuron that's basically at rest. And then they inject some current to cause the memory potential to change from -65 to -40. They're so like artificially depolarizing it, because they want to see if I change the memory potential artificially around these channels, what do the channels do? And so we're here. This is all like a time recording on the x-axis. We're going along. We're at rest. And then we inject current. And we're going to maintain the memory potential at like -40 millivolts or so. When that happens, what you see in your patch clamp recordings is that eventually these sodium channels that you're recording from will open. So here's one that opened, and it stays open for a tiny little bit of time, maybe a millisecond or so. And then it closes, and it stays closed. This is a different recording from the same neuron, but it's a different channel. It opened just slightly after. It didn't quite open as quickly, so there's a little more of a delay. It stayed open for about the same amount of time, and then it closed, and then stayed closed. This one opened pretty much the same time this one did, and it stayed open for a little longer than the other ones, but then it closed. And you could do this over and over and over, and you see little variations like this. But in every case, what you see is that the sodium channels will tend to open, and they'll stay open for a short while, and then they'll close. And then they will never open again. The only way they could ever get these channels to open again is if they drop the membrane back down to its resting memory potential, and then depolarize it again, and at that point they will open. So it's almost like they become inactive, and they need to reset before they can open again. So this is like the electrophysiology. That was really tantalizing because it offered some clues as to what these things must look like and how they must work. And much subsequent work was done to work on the structure of these sodium channels. And this is kind of a cartoon of how we think they actually work and why they would do this thing where they open for a short period of time and then they close. Here is a resting sodium channel. So this is when the membrane is at rest. This is how they would look. So the sodium channel is closed. You can see that. So like this is the channel part. But this gate down here is closed. This is the activation gate. And so at this point, sodium ions cannot go through this channel and get to the interior of the cell. It's locked shut. What will happen, though, is that if you change the membrane potential around them, there are certain positively charged amino acids that are in this structure of the sodium channel that will respond to that memory potential. There's a question in your homework actually that asks you about that, so I'm not going to get into that now, but I think it's really cool. When you change the memory potential around them, so you go from minus 65 to minus 40 millivolts, they will kind of change shape a little bit, and as they change shape and shift, it causes the channel to open. So now sodium ions can start rushing in, and that's what we're recording here. We've got inward currents of sodium ions rushing in. And then, inevitably, what will happen is they seem to close, but the reason they close is not because they go right back to this conformation. Instead, the reason they close has to do with this thing down here. This is called an inactivation gate. I kind of equate it to a cork on a string. And so, these things are, in solution. So these molecules are bouncing around all the time. So that means this little cork structure, this inactivation gate, it's a globular subunit, it's just labeled over here. This globular subunit is bouncing around all the time. And once you open this channel, it creates this pore that the globular subunit can fit in. And it's not really attracted to the pore, per se. It won't immediately fly into it, but it's bouncing around, and eventually it'll bounce in such a way that it'll kind of jam in there and get caught. And at that point, then, this channel will be inactive. So in these first two cases, it didn't take very long, maybe a millisecond or so for the inactivation gate to finally find its home. In this case, the gate was bouncing around a little more, didn't happen to hit, but eventually it hit, and then we inactivated. And at that point, then, this channel is kind of stuck in this conformation. The only way to get this channel to open again, such that ions could actually go through, is you have to get the memory potential to reset back down to minus 65 millivolts. When you do that, that causes the positively charged amino acids that are in the certain subunits of this channel to shift again, which will cause the channel to change shape back into its closed conformation, and that will kick out the globular subunit. And at this point, this channel is ready to open again. But it's not until you sort of reach this point that we can open the channel again. So again, this is at rest. Once the memory potential changes, this thing will open. Sodium ions rush through, and they'll keep rushing through until this inactivation gate, this globular subunit, will plug up the opening. At that point, the current stops. And you can't open the gate again until you get this channel to reset. So when it resets, you close the activation gates again. You get it back in this conformation. That ejects the globular subunit out, and now this thing is ready to open back into this shape. We talked about the absolute refractory period. That's this period of time when you cannot start another action potential until the first one has sort of passed. The thing that causes the absolute refractory period is basically this inactivation gate. So we need to get these channels to close and reset again, which is, and you would have to do this to start another action potential. You can't have that happen until you get back down into your resting memory potential, because that's when the channels reset. So the absolute refractory period, that thing that limits how many action potentials you can fire per second, it's caused by the time it takes to get back to resting memory potential and eject this globular subunit. So there's a way in which I like to say the globular subunit, this inactivation gate, is responsible for the absolute refractory period. It's the thing that's you have to reset and undo in order to get the neuron to be able to fire an action potential again. How's that? I mean, is that okay? Questions on what we're doing with sodium channels? This is how they work, these voltage-gated sodium channels. Now, these voltage-gated or voltage-sensitive sodium channels that we're talking about, these guys, so these are real structures. We've recorded from them. We understand their structure as far as how they open and close. I guess the other question would be what makes them so specific. We had the same question about potassium ion channels. Potassium ion channels were extremely specific, and these guys are too. Their structure is actually really similar to what we saw with potassium ions, with the potassium channels, rather. So again, they're composed of a bunch of repeating subunits. There's usually 4 of them. Along the interior of each of the repeating subunits is this pore loop. And so if you sort of looked from the top view, this pore loop creates basically like this little hole through which sodium ions can pass through. And there are amino acids that are around that pore that have very appropriate or have amino acid-based structures that will mimic water molecules. So along the pore loop are lots of oxygens that kind of stick off with hydrogens attached to them. And they mimic these oxygens that are normally found on water molecules that would be surrounding a sodium ion. The hole, in this case, is basically just the right size for a sodium ion and the water molecule that it might be attached to fit through. And so this is what this is trying to show, is that like this little hole that's created by the pore loops, that's called the selectivity filter. And they're sort of conceptualizing it with this little box here. The selectivity filter is just the right size for a sodium ion and a water molecule that's attached to it to breeze right on through. If a potassium ion, on the other hand, tried to go through, it would be also associated with water molecules, and the selectivity filter is just a little bit too small to allow a potassium ion and adjacent water molecules to fit through. So this guy basically just gets caught and is unable to pass through the selectivity filter. So it's kind of the same concept. The potassium channels were like just the right size for potassium to go through. The sodium ones are just the right size for sodium to go through. When they don't have the perfect conformation, they kind of get caught in the way as they're passing through these structures. And that's what allows them to be so specific. OK. So far, basically what we did is we said action potentials are caused by sodium channels opening and closing. That's kind of like the summary of the lecture so far. That is not the only thing that happens. I mean, one thing that you probably noticed is that in our really simple model, we basically we depolarized and we repolarized, but we never did this undershoot thing. And in fact, if we had any kind of scale, really what we would have done is we would have depolarized, and then we would repolarize by closing those sodium channels, but it would kind of take a long time. I mean, we wouldn't get back down as fast as the real action potential gets down. The falling phase occurs really quickly, too quickly to be driven just by sodium channels closing and then leak channels kind of taking over. And furthermore, we have no explanation for the undershoot. There's no reason the membrane potential would ever drop below the equilibrium potential of potassium, given our model so far. So we need another player. And it turns out that the other player is that we have a whole other set of potassium channels, in addition to the potassium leak channels that are involved in an action potential. And they're called slow voltage gated potassium channels. They're called slow because they just kind of take their time to get open. Like they're just not as quick and responsive as sodium channels are. They have a slightly different structure. The structure results in them just not being so eager to open. And so what will usually happen is that when you hit threshold, you open your sodium channels really fast, and then they close and inactivate up here. I should say the sodium channels inactivate. And right about the time they inactivate, you also tend to open a bunch of these potassium channels. But potassium channels, these potassium channels do what potassium channels do. They open and they allow potassium ions to flow through them. Potassium ions always flow from the inside of the cell to the outside. They're positively charged, so they make the inside of the membrane more negative because they're flowing out. They're taking their positive charges with them and make the inside of the membrane more negative. So what we're basically doing is we're kind of jump-starting the repolarization step. We depolarize by opening a bunch of sodium channels. The sodium channels inactivate right around here. And then we open extra potassium channels to allow this huge current of potassium ions to stream out. And as a result, we have a very fast falling phase. And in fact, there's so many potassium ions falling out, the membrane potential becomes so negative that we actually undershoot past the resting membrane potential, and we're down into this hyperpolarized state. So the next slide kind of shows this. I don't have time to go through the whole thing. The point is, and we'll come back to this at the beginning of class Friday after the quiz, that the rising phase of an action potential is all due to sodium channels opening. The falling phase is when the sodium channels are closed, and then we have this extra potassium efflux, extra potassium current coming out. So we'll walk through this figure on Friday. Sorry I went over. I will see you guys then.